This talk is going to be not a critical review of what has been done in the field of microstructure imaging and machine learning in diffusion MRI, but it's rather an overview of the work we have been doing in Cardiff University and Kubrick, and the work that my team and I have been delivering in the microstructure imaging. So it's very much an explanation of the work we have been doing. And the central topic is the microstructure imaging. And so I believe during this workshop, you have already been exposed to the concept of microstructure imaging and what diffusion MRI is capable of. So I will start with just a quick introduction, and then I'll show you a series of examples of where we can push this idea of mapping tissue microstructure using diffusion MRI and computational modeling. So the grand challenge that we try to solve is to use large-scale signals, so signals that we acquire using magnetic resonance imaging, on a large scale, which is the millimetric scale of your image voxel. And inside this millimeter cube voxel, you can have a very complex microstructure. So the structure of the tissue at the cellular level. That's what we mean for microstructure. And here is an example of the kind of contrast in a T2-weighted image, the B0, you would see, and the underlying tissue that generated this contrast. So in the white matter, you might see something a little bit different, and the microstructure underneath it is also different. So how can we go from something that we measure at the macroscopic scale down to features that are microscopic, so micrometer scale? There are many different ways using MRI we can do this. We can bridge this resolution gap. And one of the successful approaches proposed more than 25 years ago is to use the properties of the displacement of endogenous water molecules within the tissue to obtain an indirect information of what is the cellular membrane arrangement within your voxel. And so here the basic concept is that if we were able to label water molecules diffusing within the tissue, looking at the displacement of these water molecules over time, we should be able to also infer some of the properties of the microstructure. So here I give you an example of how this displacement of many water molecules would look like if they were hindered in their diffusion from the axons in the white matter. So the axons are all aligned in one direction, let's say the Z direction here. And so the molecules are more free to displace to perform longer displacements in the direction of the fiber rather than orthogonal to the fiber. So let's say diffusion is more free in this direction and more hindered in the orthogonal direction. And so if we look at the probability density function of this molecule to perform a particular displacement, it has interesting properties. It is anisotropic. And not only we can tell that it is anisotropic, we can also identify the main direction of a free diffusion. In the gray matter is a little bit more complicated because the arrangement is very random and isotropic, so we have membranes all over the place. And so the displacement itself looks quite isotropic. So if we want to map or obtain interesting features of the microstructure, we need to do we need to make some assumptions, and it's a little bit more complicated. So in what I have said, the central piece is that we need access to the probability density function of water molecule performing a displacement from position R to position R prime in a given time tau, which is what is also called the diffusion propagator, and which is what I call the G in this equation. And there is a nice relationship between this probability density function and the signal that we measure with diffusion MRI. The signal is the Fourier transform of this probability density function. Now, the question of accessing microstructure can be reformulated as can we obtain information about this probability density function? And because it's the Fourier transform, there is this term here which allow us to probe this probability density function. Let's have a look at how this works. So this probability density function for diffusion MRI comes from the solution of the diffusion equation. And if you would like to solve analytically this equation for any given geometry omega, you need to know what are the boundary conditions to this differential partial equation. And here is where most of the problem comes, is because we need to know what is the boundary that we are considering. And we need to also define this boundary in a convenient way to then solve this equation. Historically, this has been done using simple geometries for which the boundary condition is easy to write, can be written easily, and the equation can be solved in a straightforward way. And so here you see an example of diffusion between two parallel infinite planes for which we know what is the boundary, is two planes at a distance 2a. And then we assume perfect reflection, so Newman condition at the boundary. We solve the equation and we obtain this solution for the propagator, for G. And for spheres, likewise, you can impose these particular simple geometry, use the spherical symmetry to solve the equation, and you obtain this solution for the propagator. Once you plug this solution inside this equation, so you calculate the Fourier transform, you can have a first approximation of the signal that you measure. Once you have access to the boundary, once you have defined the boundary, the second aspect is how I'm going to probe the properties of this function. Because, okay, I know how it is analytically, but also I need to tune my acquisition to extract information from that equation. And this can be done tuning the parameters of your acquisition. So typically, if you use a pulse gradient spin echo, PGSE, that's the kind of scheme. You have a 90 and 180 RF pulses, and then you have a couple of diffusion sensitizing gradients. And here you have pretty much 3 degrees of freedom. You can change the separation between these two pulses, and this separation changes the time scale at which you probe the propagator, or if you wish, it dictates how long you wait before you start encoding the displacement from R to R prime. And then you have the gradient strength and the gradient duration that enters into the Fourier wave vector. And so you can probe different, let's say, spatial scale when you change the gradient strength and the small delta. So with these two, let's say, with this arsenal of tools, one is simple geometries for which we can calculate the diffusion propagator, and the other is a sequence that allows us to probe this diffusion propagator. Then we can start doing microstructure imaging from a bottom-up modeling. And this is what historically has been done in the past 20 years in the field. And you might have seen some examples in this workshop. And so usually you acquire the data. So here we are changing the gradient direction and the gradient strength. And so we are tuning our acquisition to be sensitive to the diffusion propagator. We normalize the signal in each voxel so that then, looking at voxel by voxel, we can have a signal decay as a function of, for example, the diffusion weighting. And now it's this signal decay that we are probing that we want to link to the microstructure. And here is where we make some strong assumptions that are needed to solve analytically this, to extract analytical solution for the diffusion propagator. So when it comes to the white matter, we assume that the axons are straight, infinite long cylinders with a certain radius, hollow, so the water can diffuse inside and outside. So this is a strong assumption, strong modeling assumption. However, it allows us to use the analytical expression that we can calculate given our acquisition And then we need to build a model for the tissue. This is the model for the individual components, the axons. The model of the tissue can be written as a weighted sum of many of these elementary compartments with different sizes following a given distribution, in this case, age. And so once we have this model, this is all analytically well defined. We can easily use nonlinear least square optimization approaches to fit this model, signal, to the data and extract in each voxel an estimate of our model parameters. And here there is the second big assumption we are doing, which is our model parameters are a realistic representation of the actual tissue parameters we are interested in. So what we can estimate is cylinder radius from our cylinder model. And then we say this cylinder radius correlates highly with the axonal radius. So this is a statement, this is an hypothesis that needs to be validated. And so a lot of the effort in the past 20 years has been also put by the microstructure imaging community in validating with histology this statement regarding axonal radius, axonal density. Then we moved on into neurite density, neurite dispersion, and now recently in the gray matter, we have Soma size, Soma density, and so on. So each time we build a simplistic representation of the tissue, that is a model, and the model parameters can be estimated, and that's for sure. But then how they link to the underlying biology still needs histology to be
fit a
validated.
model that is
now, what we tried to do is to move away from these strong
24 channels
simplification of the model. And we thought if we need to use a simplified model, we need to identify what are the relevant features. And this often is done with a priori hypothesis based on theoretical considerations. However, nowadays we have tools like a lot of electron microscopy data and a lot of high-resolution microscopy imaging that can help us identify these relevant features. But to do so, we need also to understand what of these features can induce a significant change in what we measure. So here you see an example from some of our work a few years ago, where we took a complex three-dimensional structure mirroring the neurons in the cortex, and we performed the diffusion of many molecules in this structure. And then we simulated using Monte Carlo our diffusion weighted signal and estimated what would be the apparent diffusion coefficient coming from such an experiment as a function of time. And we did this for many different cell types. The first thing we observed, without going down to the individual features, is just that each cell type in the brain seems to carry a specific microstructural fingerprint. And so here is where we start thinking of this microstructural fingerprint as an HistoPrint that we could link to particular histological features, such as the Soma size, which is different between these three neurons, the branching order, how many cellular projections bifurcate, which is different between these three neurons, and the length of each of these branches, which is different. So these are all features that we can now not only hypothesize are relevant features, but we can also investigate with our simulations. And so we embarked in the last 5-6 years into the handover to generate simulators that are more and more realistic in order to interrogate them and to use them to help us identify which one are the features that we can actually quantify. And so when it comes to white matter, not only us, many other groups as well have come up with their own generative model of computational representation of the tissue. Nowadays, we could, once you link this to specific individual bases, so you try to fit this model at the individual subject, you can also call them digital twin, because it's a digital representation of what the microstructure in that voxel for that person
in the brain
should look like.
And this
the real challenge for the white matter, nowadays there are tools available to simulate with some realism the individual axon. The real challenge was the gray matter, and still is the gray matter. For gray matter, we are missing accurate, morphologically accurate simulator of the full complexity of the gray matter tissue. And so I will show you in this talk some advancement we have done in Kubrick about this challenge and how we think we are at a good point to address it. Obviously, you might have seen also during this workshop, nowadays the machine learning and deep learning is a very powerful tool to perform a model parameter inference. And so at the same time, once you have access to a computational model, which is also a generative model, you can imagine to generate many random realization of microstructures and then use it to train a deep learning model to try to infer what is the most likely microstructure that give origin to the signal you measure in each voxel. And so following this philosophy, I'll show you also examples of how we are able to do this mapping at the moment. And I'll show you some application in the white matter, demyelination, and cancer. So for what it follows, I'll just dig a little bit more into the idea of the computational modeling and how you can train models to then perform the inference. So initially, almost 10 years ago, we proposed one of the first application in MRI, where we used a three-dimensional graph to represent the neurons. And we built a generative model that accepts as input a few microstructural parameters, which are the number of projections radiating from the soma, the length of the branch of each projection, how many times they bifurcate, and the diffusion coefficient of the molecules we want to probe with the diffusion MR. And then if you feed this into the model generates copies of thousands of cells, individual cells, all respecting these particular priors on their morphology. And once you have these cells, you can simulate 100,000 spins diffusing inside of each cells with a specific diffusivity. And you can start building a dictionary of these HistoPrints. So the imprinting of the cellular morphology onto our measured diffusion signal. In this particular work, we didn't apply this directly to the diffusion signal, but we applied it to a surrogate matrix from the diffusion signal, which is the apparent diffusion coefficient. So we simulated the full signal, and then we fit the slope of the log of the signal at the low B to estimate this ADC at different diffusion times. And so we have a dictionary of matching pairs of parameters, set of parameters for our model, and simulated ADC signal. We can train a machine learning model. In that case, we trained a random forest to learn from a real signal then what would be the most likely combination of parameters that originated it. And here you can see an example of the data. The real data are the dots, and then this is the fitted curve. And we have done it for different molecules. So with diffusion MR, we can not only use water, but we can also use other molecules. We can encode the signal from other molecules other than water. Why is this convenient? It's convenient because some molecules, such as the N-acetyl aspartate, they are mostly inside neurons, while inositol or choline are mostly inside glial cells. And so this can give us also a cell type specific information. And second thing is that these molecules are purely intracellular. So we don't need to simulate the whole tissue. We just need to simulate individual cells. And then the total signal is the volume weighted sum of the signal from each individual cells. If we would use water, we need a model for the whole tissue because water is inside and outside the cells and perhaps can also cross the membrane. So back then, we simplified a lot the measurement, the modeling, but it required some sophisticated diffusion-weighted MR spectroscopy protocols to acquire this data. Anyway, the bottom line is that we were able across species to show that we could estimate the most likely cellular microstructure originated the signal in very good agreement with histology, not only at the individual species level, but also across species. So the molecules that should provide information about glia indeed gave us a ratio of 2.5 between the glial cells in the macaque compared to the mouse, in particular the astrocytes, which are those occupying most of the volume. While for neurons, we didn't find any difference. Later on, Clémence Lignol adopted this approach to investigate how well we could characterize astrocytic reactivity in an animal model called CNTF, which induce massive astrocytic reactivity within the mouse brain. And here again, we were able to show also with direct comparison with real microscopy data from the same animal that we were able to capture the most relevant features of the cellular morphology, in this particular case of the astrocytes. And here you can see in dark and light the comparison with the histology of some of the features we estimated, such as the soma size, the cellular projection size, and the branching order. And in a very recent work, always with Clémence Lignol, we applied this approach to try to decode the neurodevelopment in mice. And here we targeted two regions, the cerebellum and the thalamus, that we know undergo two different neurodevelopment pathways from 5 days post birth to up to 30 days. And here you see how with this approach, assuming that most of the signal coming from the voxel comes from Purkinje cells, for example, you can start following how with neurodevelopment, these cells become more complex and more branched. And in the thalamus is big pyramidal neurons. In the paper, there is much more than this. This is just a snapshot to give you a feeling, but feel free to have a look. There is also a preprint out. However, so far, this seems very encouraging, and the results align very well with the microscopy and even quantitatively. However, we have used a quite simplistic model for the cell, and we haven't done the tissue. We have done only the cells. So can we do better? And that's where, with my team, we have been working a lot in the past 4 years. Obviously, to address this challenge is not that simple, and you need to work step by step. So first of all, you need to define a better model for the individual cells, rather than 1D graphs embedded in a three-dimensional space, having actual meshes and a generative model that can capture modulation, caliber variation, and all these subtleties that the previous model wasn't encoding. Second, you need a way to pack these cells together. And I don't like to say pack, I prefer to say grow, because I think nature don't work in the way that takes the cell, put together and then try to pack them. Nature actually grows cells contextually. And that's how I will show you example of how we manage now to grow individual cells and contextually together. Then we need a simulator, for which luckily in our field, a lot of groups have produced very, very good simulators. And then we need a way to do the mapping. Okay, even if you have paired sets of signal features and microstructural features, how do you do this mapping in a reliable way? And so not only the estimates of these microstructural features is important, but the uncertainty around it is extremely important, because the problem we are trying to solve is ill-posed and very often is degenerate. So we need ways to estimate, at least to characterize this degeneracy. Finally, we need always the histology. We can't use this approach at the moment in an exploratory way. This can only be used in combination with histology because of what I said at the beginning. What we estimate in the end will be model parameters. They are not tissue parameters. And then we need to reassure with strong correlation with the actual histology and the actual tissue that what we measure is not far from reality. So I'll show you example of what we have done in the first to pack and create this tissue. So everything started a few years ago, where we identified from an investigation of the histological reconstruction 12 morphological features that we believe are important for diffusion MR. And these are based on theoretical considerations. The second question was, okay, how do I learn for each cell type? How do I learn these features? How do I ensure that my model will learn this feature and then create new neurons? So here the idea is to train a generative model that learns from microscopy how different cell types look like in 3D. So Purkinje cell, motor neuron, and pyramidal neuron, for example. And then once it's trained, we can generate millions or as much as we want copies of these neuronal populations. And we can use it to train a second model, in this case, a deep learning model, to do the inference. And so here you see an example of how back then we used an heuristic approach where we downloaded the real cell reconstructions. So this is an example of 7 motor neurons. And then we needed a matrix to optimize. So we need a loss function to minimize when we want to learn, when we want to train our generative model. And so in this case, we used the persistence image introduced by Lida Canari, which is a very elegant and convenient mathematical framework to characterize complex morphology of neurons. And in this space, you can see that heuristically, changing randomly the combination of the model parameters, simulating the cellular structure, and calculating this loss function in an iterative way, we converge towards a minimum that allow us to create copies, random copies, of the original cells. So these are not exactly one-to-one. These are statistically equivalent morphologies to the target ones. How do they look like? They also, obviously, they have to look morphologically similar. And so here you see an example of the obtained synthetic cells. What we are interested in is not just the morphology, it's actually the signal that we simulate. Is it realistic enough? And so if you compare the signal from Monte Carlo simulation you get from Purkinje real one or fake one, they overlap basically. And within also, this is also noise free. If you add the noise, they are basically identical. So this means this test, and there are other tests in the original paper, mean that we can use this generative model to create morphologically accurate copies of each cell type. But this also means that now I can use my model to do what I mentioned at the beginning, try to understand which features of the cellular morphology do impact the measured diffusion signal, and can we then design an approach to measure each of these features. And so here you see an example where I remove or add features to the morphology and how the final neuron look like. The first thing that or at least we need to start with one feature. So the first feature that to us came obvious to explore was the Soma, was the cell body of the cell. So what happens if you have it or don't have it? Does it change the diffusion MRI signal? And if it does, how it changes it? Changes it in a way that is unique, and so we can try to separate it. And this was the beginning of the initial work that then led to a model that we called the Soma Neural Density Imaging, or SANDI. In the original paper, again, you can find more details about this, but the basic idea is that using numerical simulations with those generative models, we were able to show not only that the signal coming from the Soma has a peculiar signature in diffusion MRI compared to the signal coming from neurites, but also that if you use simplified models instead of the actual meshes, you get a very good approximation of the signal. So you can simplify even further and propose an analytical model, a compartmental model that actually match very nicely the full simulation of the cell. And this is what SANDI is an analytical model that has been applied and reproduced by different labs. And one of the interesting aspects is that allows you to estimate Soma compartmental signal fronts. And then we showed qualitatively that there is a match with histological staining for Soma for cell bodies. Then follow-up work with Andrada Janusz demonstrated more quantitatively that the sphere fraction from the SANDI model correlates with the actual cell body density in the Allen Brain Atlas. And then ongoing work in collaboration with Noam Shemesh tries to map more one-to-one what we measure with our model and what is the reality. So here you see an example in the cortex of a rat, of a mouse. The expected layering. Here each yellow dot is a cell body. And you can see how some layers have many more cell bodies than others. And on the same mouse, you see the estimated model parameter from SANDI. And so you see that there is a strong correlation across the layers between what we estimated and what is the reality. So obviously it's not one-on-one. So here, if you walk across this cortical layer, what we estimate with diffusion MRI don't have the exact same number of what you estimate with DIPY, but simply because it's two different ways of measuring. And that's why it's important, the correlation in this case. A very recent work that Eleonora Lupi is going to present at the next ISMRM implies the increasing the resolution of these approaches, because it would be really amazing if we could quantify this layering in the human cortex in vivo, but we have a limitation, which is the resolution. So what Eleonora did is to adapt a machine learning approach called the image quality transfer to work for data acquired at ultra high B value and at the different diffusion times. And she was able to show, with comparison with the big brain histology dataset, that we were able, super resolving up to 500 micrometer cube, we're able to resolve features in the brain cortex which are only visible on the big brain. And if you use the original resolution, which was 2 millimeter isotropic, you don't see those features. So there are two nice results here. One is that actually what SANDI estimates correlate nicely with the actual cell density. But the other result is that it's important to push for resolution when you want to use these kind of techniques. Because in the cortex, you really want to try to see all those features. Okay, then here is just a summary. If you're interested, SANDI has been quite used in many different aspects. And so if you are interested to dig more into the application side, I recommend these recent works. How do we grow the systems? How do we grow the tissue? Okay. How do we grow the tissue? This is the work of Charlie, really. So whatever I'm going to show you now, which is one PhD student in our group, has been entirely developed by Charlie. And so here the idea is to grow cells, but we want to grow them morphologically accurate. So we want to end up having morphologies of the individual cell and the full tissue, which are a perfect mimic of the microscopy, what we see in microscopy. So the idea is to use a pseudo-random generator where we have a cell body, the red dots, and then the branching and the projection start growing and branching in space randomly. But they need to follow some rules, and we need to learn these rules from histology. So what Charlie has done has been taking thousands and thousands of reconstruction from open source databases and explore each aspect of each cell type of morphology. We have done it for 9 cell types, 3 glial cells and 6 neuronal cell types. In addition to that, we borrowed again from Lida Canari's work convenient mathematical representations that can help us encode these features and then call them back into our loss functions. And finally, for each cell type, you also need to understand towards where each fiber needs to grow. Because if you take pyramidal cells, for instance, you can see they are very polarized. They grow from bottom to top, for example. But if you take astrocytes are a little bit more isotropic. And so we need this information. And this is what you see here in the red dots is what we call attractor points. And so once you start on the Soma, how many branches you need to have is learned from the microscopy, from one of these distribution. And then once you take one of these fiber, towards where this fiber should grow is randomly selected from the distribution of attractor points learned from microscopy. The rest is all completely random. And you can find all these descriptions in a recent preprint that Charlie has uploaded, and we are working at, obviously, at the extended manuscript to be published. But you find all these details there. And so here you see some examples of growing contextually 9 bipolar neurons where they avoid each other. Or here is when we grow 6 astrocytes where they avoid each other. But you can do much better. So here Charlie grew 288 pyramidal cells avoiding each other. Each of them are all drawn from the same family. So they all are statistically the same, but equivalent, but they are not morphologically identical. And here we demonstrated last year at ISMRM that the final results, so in red is the real cells from the reconstruction, and in orange is our generative model, mirrors very well the distribution of features when we grow them individually. And this basically confirmed the results we obtained in 2019. But most interestingly, contextually. So if you grow them together, they end up being morphologically accurate because we have encoded it in the growth. And Charlie since then has started playing mixing cell types. So here you see an example of pyramidal neurons and astrocytes, same story, very accurate. And finally learned the distribution of the cortical neurons. And so we picked up the neurons, we arranged them into layers as it should be, and fed it into our algorithm, which is called CONSEG, and obtained a full core mesoscopic cortical column of neurons. This is 1.2 millimeter times 200 by 200 micrometers. So it's really mesoscopic. We are really simulating now full voxels or multiple voxels. And here is an example of neuronal and astrocytic cell bodies. And once again, the signal that we obtain is from this synthetic digital twin and the real one overlap perfectly. And so this suggests we can now use this for the 2 purposes. One is explore the features, and second is to build dictionaries to train deep learning models. And you can see how we can use it to break out voxels that are the same size of a preclinical MRI, and we can start then interrogating extracellular, and so on. Moving forward, Katir Shimshek and Ioana Derukaki in our team took this cellular microstructure, which encapsulate a lot of the complexity, but not all. There is something that we were not encoding in our algorithm, which are spines, which are these little lateral protrusions in the dendrites. And we believe that these spines carries a significant and unique signature in the diffusion MRI signal, so we can measure them. That's the idea. So we started with simulations to show if our hypothesis was right. And in short answer is yes, it's right, you can. And then we start working towards how to optimize the acquisition to measure spine density. And so here it was the preliminary work. We proposed a phenomenological model, which was anyway introduced in the 90s by Jablonski and Sostansky. We extended it a little bit for spiny dendrites. And the idea here is that this model has two parameters, the intraneurite kurtosis and apparent diffusivities. And if you have spines, the kurtosis that you measure should go up and the diffusivity should go down because they introduce non-Gaussianity and delays the diffusion along the fiber. But we use numerical simulations. We built ad hoc computational models that the effect of the spines at different spine densities, and we showed that there is a significant. So this is the kurtosis, the intraneurite kurtosis versus spine density, as you can see for different acquisition schemes. So the blue one is preclinical. So if we have access to a preclinical scanner, we can already measure with a very good sensitivity spine density. That's the idea from the simulation. If you move towards clinical, it's more the green one. And so let's say the green for a very good clinical scanner, otherwise you are in the red. So in the red, it means you don't have much sensitivity, so it would be challenging. But for scanners like Prisma or the SIMA-X that are coming in the market, you are between the green and the blue line. So here there is great potential for exploring this kind of mapping. And here we have done some analysis to show how sensitive you are to changes in spine density, because some pathologies or conditions, they lead to increase or decrease of spine densities. And so it seems once again, with the preclinical scanner, we are already there. With clinical, you need to have a very, very high SNR. So it's still a bit challenging. But again, Connectome scanner, SIMA-X, they will go into this blue regime. So really exciting years to come where we can really apply this to human. We already have done it with a Prisma in collaboration with Francesca Branzoli, and we targeted cortex and cerebellum, where we know in the cortex we have half the spine density than in the cerebellum. And so we measured the metabolites, so we can make the modeling simpler. This is the NAA in the two regions, and you can see that the region, the cerebellum with the higher spine density, had the higher kurtosis compared to the other. You can use our simulation as calibration curves. What would be the spine density that give us these kurtosis values? And we found out that it would be 2 spine per micron for the cerebellum and 0.81 per micron for the cortex, which is exactly what is known from histology. So this is, again, a very encouraging first results. What Melis Jallet, postdoc in our team, has done is to bring this to preclinical scanner, use double diffusion encoding, for which she demonstrated that there is higher sensitivity compared to single diffusion encoding to spine density, and then apply this in collaboration with Julien Valette lab to an animal model called APPS1 of neurodegeneration. And we were able to estimate what our model says is a 67% spine loss and a 250% glial activation, increasing glial volume. And at least for the spine loss, we know that in this animal model, we expect 60% decrease. So we were very happy with these results. And obviously, we need now to do the logical validation about this. This will be presented by Melis at this year ISMR. And I think Alice herself has already covered on Tuesday in this workshop how we plan to estimate uncertainty. So I will not go over this. I think Melis did a great job last Tuesday to present this. So I encourage you to watch the video if you have missed it. And I want to just conclude with a quick show, like 5 minutes quick show of what we have done in applying this approach. So we came up with the generative model, computational models for cancer. And here you see an example for glioblastoma. For white matter, and this is the work from Ross Callahan and Config. And then the work from Charlie will push the boundary for the gray matter. So using CONSEC and Ross' work, we were able to simulate a large set of different pathologies for white matter. So here an example starting from the original, this is the other way around. This is the original. And then here you have demyelination. And here you have, this is the original. Then you have only axonal loss, you have only demyelination, or you have both. And then you can tune with this. So we did this, we trained the model to recognize demyelination. And then the model, so in this case, again, it was a random forest, simple, just to show that it works also without fancy deep learning. We were able to show that in Kuprison mice, the demyelination was obvious and was quantitatively correlating with the actual myelin thickness estimated from electron microscopy. And this was a work from Joanna Hill. Then Eirini last year presented at ISMRM our approach of highlighting the pathology in epilepsy. And here you see how to take neuronal reconstruction, and then we artificially create balloon cells or abnormal neurons that are typical of epilepsy. And then we simulate the diffusion signal, and we ask what is the best acquisition to identify tissue damage. And this is not high b-value, so quite standard. But what if we want to classify the subtypes of epilepsy? Then you need ultra strong gradients. And what else we did is training the machine learning model to then estimate what is the fraction of balloon cells and dysmorphic neurons in each lesion. And here you can see how, in this particular case, because there are more dysmorphic neurons, this was a type 2B, which was confirmed by the histology. In cancer, we have done the same. We have used machine learning and numerical simulations to source separate what are the different contributions to measure the apparent diffusion coefficient. And so you can now estimate what is the typical diffusivity, cell radius, and volume that can lead to the measured ADC. And this was used in a center in Milan to assess treatment response in meningioma. And here you see that thanks to our source separation algorithm based on numerical simulations, we were able to clearly distinguish the people who were at high risk to not respond to treatment compared to who was responding. And this was done also from the same group a couple of years later in Chordoma. And here you see a striking example. With the ADC, the original measurements, you were not able to stratify your patients. So red is who didn't respond and green is who responded. But if you use our source separation method, you were able to statistically discriminate respondent from non-respondent. So this is an example of how our models can also enhance the analysis of already acquired data. And finally, this is application to prostate cancer, where we developed a new model, which is combined diffusion and relaxation. And we showed that we were able to discriminate also the most aggressive Gleason score, so the cancer which are more clinically relevant. And no other technique at the moment is able to do this. And this was informed by numerical simulations. And now what we are doing is using all these model parameter maps to train a CNN to generate a cancer probability map, but also a virtual Gleason scoring. And the preliminary results are quite encouraging in this direction. So with this, I want to conclude, and I tried to give you an overview of the work we have been doing on computational modeling and how we believe this can be combined with microstructure imaging to overcome some of the limitation of the current works. There are still, it's still essential to have histology, actual histology, and the estimation of the degeneracy and the parameter uncertainty is extremely important, because what we are trying to do is a nilpost problem. But this talk combined with Melly's talk on Tuesday should give you a good overview of how we believe we have all the ingredients now to start addressing this challenge. And with this, I would like to thank you all for the attention, all the collaborators that I mentioned during the presentation, and the micro team at Kubrick.